Not all ligands for the T-cell receptor produce a similar response. So far, we have assumed that all peptide MHC complexes that are recognized by a given T-cell receptor will activate the T-cell equally. That is, that the T-cell receptor is a binary switch with only two settings, on and off. Peptides that trigger the on setting are called agonist peptides, by analogy with the agonist drugs that activate other types of receptor. However, experiments originally designed to explore the structural basis for antigen recognition by T cells unexpectedly showed that the recognition does not necessarily lead to activation. Indeed, some variant peptide MHC complexes that do not themselves activate a given T cell can actually inhibit its response to the agonist peptide MHC complex. These peptides are usually called antagonist peptides, as they antagonize the action of the agonist peptide. Other peptides that can trigger only part of the program activated by the agonist peptide are called altered peptide ligands or partial agonists. Such ligands can, for instance, induce the lymphocyte to secrete cytokines but not to proliferate. Most of the agonist and partial agonist peptides that have been described are closely related in sequence to the agonist peptide, although this is not always the case. Several studies have suggested that the binding interactions between T-cell receptors and these variant peptide MHC complexes are of a lower affinity than the binding interactions between a T-cell receptor and its agonist peptide MHC ligand. The extent to which agonist peptides and altered peptide ligands influence physiological immune responses is not known, although they might contribute to the persistence of some viral infections. For example, mutant peptides of epitopes on cells infected with human immunodeficiency virus, HIV, can inhibit the activation of CD8 T cells specific for the original agonist epitope at a 1:100 ratio of mutant to agonist. This sort of effect could allow cells infected with mutant virus, which arise commonly during the course of HIV infection, to inhibit virus-specific cytotoxic T cells and thereby promote the survival of all infected cells. Differential signaling by variants of agonist peptides is thought to be important for the development of T cells in the thymus and their subsequent maintenance in the periphery. T cells are selected for their potential to be activated by foreign peptide antigens in the context of self-MHC molecules through interactions with self-peptide, self-MHC complexes. This process seems to require the receipt of survival signals from self-peptide, self-MHC complexes that are related to, but not identical with, the foreign peptide self-MHC complexes that the mature T cell will recognize. The basis of the incomplete activating signal delivered by the altered peptide ligands is unknown, but it has been shown that the recognition of these ligands leads to altered phosphorylation of CD3 epsilon and zeta-chains, and to the recruitment of the inactive form of ZAP70 tyrosine kinase to the T cell receptor. Cross-linking of the T cell receptor alone without any co-receptor engagement can also generate the same partial phosphorylation events within the cell. Thus, the incomplete signal generated by altered peptide ligands might reflect a failure to recruit the co-receptors or a failure of the T cell receptor to interact with the co-receptor productively. Also, because the affinity of the T cell receptor for the altered peptide ligand MHC complex is lower than that for the activating complex, the T cell receptor might dissociate too quickly from its ligand for a full activating signal to be delivered. Another possibility is that the conformational changes in the T cell receptor can contribute to the lifespan of the signaling complex and its ability to recruit LCK, and that altered peptide ligands fail to trigger these conformational changes. Antagonist signaling also recruits the tyrosine phosphatase SHP1 to the T-cell receptor signaling complex, where it can dephosphorylate the signaling complex. This recruitment, which is seen late in activation by agonist peptides, occurs within 1 minute in T cells recognizing antagonist peptides. And that's not all ligands for the T cell receptor produce a similar response. Have a great day.